Sourdough starters are mixtures of water, flour, and wild yeast. As the name suggests, a starter is used to start the process of making bread. Oftentimes, they are passed down like family heirlooms. Every starter tastes different due to the unique microecology within the area where it was made. You'll find more than just yeast in a starter. It's also the home of bacteria, fungi, and any other local microorganisms that decide to stop by. But don't be grossed out, because that's what gives it its flavor. The yeast in the mixture will feed on the flour and release ethanol and carbon dioxide. This is an anaerobic process known as fermentation. The carbon dioxide bubbles, and this is what causes the starter to rise. So the purpose of our project was to observe how the microbial communities in sourdough starters would react to stress induced by varying sodium chloride concentrations. We hypothesized that the sourdough starters would sort of adapt to the salt. Of course, the starter itself is not an organism, but we wondered if the microbial communities within the starter would react to the stress and become acclimated to it. To test this, each person in the lab was tasked with putting starters in varying concentrations of salt. Each week, we would analyze how much the starter grew and then sequence DNA samples to see what organisms were prevalent. So in the bottom left here, you can see some examples of the time-lapse imaging, as well as sequencing results. So in the rising rate experiments, you can see in figure 3 that the first question we wanted to answer was how do sourdough starters react to different salt concentrations? This data served as a foundational benchmark for us to base the subsequent experiments off of. The results from the data are interesting. Salt concentrations under 0.5 molar result in higher growth peaks than the control, but lower final volumes. Salt concentrations between 1 molar and 1.5 molar had lower growth curves than the control, but had higher final volumes. Concentrations equal to and greater than 2 molar had poor growth. These results suggest that the optimal concentrations to do further testing would be between 1 and 1.5 molar sodium chloride, as the growth curves show underperformance, but are not overly stressed. So here in figure 4, we are looking at the growth curves of starters that have been exposed to salt and those that haven't for 1 week. If the two growth curves that with added sodium chloride were close to the curves with water, that would suggest that the sourdough starters were adapting to the salt stress. As can be seen with the figure from the entire course, the starter does not seem to adapt to salt. If you recall from rising rate 1 in figure 3, the 0.5 molar starters seem to grow better than the control. This is still the case. However, the two curves that have been exposed to sodium chloride show less growth than the water-exposed curves. They do end up with higher final volumes, but again recall that this is a trait we already expected as we saw it occurring in rising rate 1. So the personal data does not match the overall results. Figure 4a suggests that the control starter trained with 0.5 molar sodium chloride actually grew the highest. It's normal to see some natural variation in experiments like this, so normalizing across the entire course allows us to see these trends. So in figure 5, we can see the growth curves after a second week of exposure. Here we are still looking for evidence of adaptation. The personal data in 5a shows that the salt-exposed starter still does not compete. The salt-exposed control does seem to perform better, but it's still beaten by the control with water exposure. The effects are even announced in the collective course data shown in 5b, where you can see the salt-exposed starter has significantly less growth than the ones exposed to water. Interestingly, we can still see the recurring phenomenon where salt-exposed starters actually have higher final volumes despite not growing as well. This is evident with the two control starters. So DNA sequencing allows you to identify the sequence of a specific biological sample. In this project, we did ITS sequencing, which analyzes a specific sequence known as the internal transcribe spacer. This sequence is found in lots of fungi and bacteria and is useful for identifying specific fungal species in a sample. Since we're interested in the specific fungal species within the sourdough communities, ITS helped us narrow this search down. We picked colonies from streaked plates, then used PCR to amplify the ITS sequences. Gel electrophoresis was used to confirm this, and FinchTV was a software used to obtain sequence info from the files. We then did a BLAST search to determine what species these samples came from. BLAST is a database that allows you to compare a DNA sequence to known sequences in order to identify what organism your sample might belong to. As you can see from figure 7, The PCR was successful and I was able to sequence several of the colony samples. Sample 2 was not able to be sequenced, but I predicted it may be Pichia crujiasvesi, based on similarities in morphology that were noted by several of my colleagues. From the ITS splash results, we found various changes in the microbial community of the starters over time. The initial starters were found to contain S. cerevisiae, Pichia crujiasvesi, Pichia occidentalis, and S. bayanus. Interestingly, both the starters exposed to salt and the ones exposed to water for 2 weeks endured similar changes in their ecological communities. In both cases, the 2 Pichia species were no longer present in the ITS samples. Also, in both cases, a new fungal species showed up, Kazakhstania humilius. In the future, we would like to continue exploring several of the findings within this experiment. Surprisingly, we did not notice any significant adaptation to the salt stress, but it would be useful to try longer trials in the future. Due to the time constraints associated with the course, we were only able to measure growth for 2 weeks, so it may be necessary that longer experiments are what will help produce this response. Another aspect of the project that we may want to address in the future is the salt concentrations. In this experiment, we held the concentrations constant, but it would be interesting to see how incrementing the concentration over time may influence adaptation and/or changes in the microbial communities.